4.5 The cytoskeleton. By the end of this section, you will be able to do the following. Describe the cytoskeleton, compare the roles of microfilaments, intermediate filaments, and microtubules, compare and contrast cilia and flagella, summarize the differences among the components of prokaryotic cells, animal cells, and plant cells. If you were to remove all the organelles from a cell, would the plasma membrane and the cytoplasm be the only components left? No, within the cytoplasm, there would still be ions and organic molecules, plus a network of protein fibers that help maintain the cell's shape, secure some organelles in specific positions, allow cytoplasm and vesicles to move within the cell, and enable cells within multicellular organisms to move. Collectively, scientists call this network of protein fibers the cytoskeleton. There are three types of fibers within the cytoskeleton: microfilaments, intermediate filaments, and microtubules, figure 4.22. Here, we will examine each. 112 Chapter 4, Cell Structure. Access for free at openstax.org. Figure 4.22 Microfilaments thicken the cortex around the cell's inner edge. Like rubber bands, they resist tension. There are microtubules in the cell's interior where they maintain their shape by resisting compressive forces. There are intermediate filaments throughout the cell that hold organelles in place. Microfilaments. Of the three types of protein fibers in the cytoskeleton, microfilaments are the narrowest. They function in cellular movement, have a diameter of about 7 nanometers, and are comprised of two globular protein intertwined strands, which we call actin, figure 4.23. For this reason, we also call microfilaments actin filaments. 4.5. The cytoskeleton. 113 Figure 4.232 Intertwined actin strands comprise microfilaments. ATP powers actin to assemble its filamentous form, which serves as a track for the movement of a motor protein we call myosin. This enables actin to engage in cellular events requiring motion, such as cell division in eukaryotic cells and cytoplasmic streaming, which is the cell's cytoplasm's circular movement in plant cells. Actin and myosin are plentiful in muscle cells. When your actin and myosin filaments slide past each other, your muscles contract. Microfilaments also provide some rigidity and shape to the cell. They can depolymerize, disassemble, and reform quickly, thus enabling a cell to change its shape and move. White blood cells, your body's infection-fighting cells, make good use of this ability. They can move to an infection site and phagocytize the pathogen. Link to learning to see an example of a white blood cell in action. Watch a short time-lapse video of the cell capturing two bacteria. It engulfs one and then moves on to the other. Click to view content, https://www.openstax.org/l/chasing_bacteria. Intermediate filaments. Several strands of fibrous proteins that are wound together comprise intermediate filaments, figure 4.24. Cytoskeleton elements get their name from the fact that their diameter, 8 to 10 nanometers, is between those of microfilaments and microtubules. Figure 4.24. Intermediate filaments consist of several intertwined strands of fibrous proteins. Intermediate filaments have no role in cell movement. Their function is purely structural. They bear tension, thus maintaining the cell's shape, and anchor the nucleus and other organelles in place. Figure 4.22 shows how intermediate filaments create a supportive scaffolding inside the cell. 114 Chapter 4, Cell Structure. Access for free at openstax.org. The intermediate filaments are the most diverse group of cytoskeletal elements. Several fibrous protein types are in the intermediate filaments. You are probably most familiar with keratin, the fibrous protein that strengthens your hair, nails, and the skin's epidermis. Microtubules. As their name implies, microtubules are small hollow tubes. Polymerized dimers of alpha-tubulin and beta-tubulin, two globular proteins, comprise the microtubules' walls, figure 4.25. With a diameter of about 25 nanometers, microtubules are cytoskeleton's widest components. They help the cell resist compression, provide a track along which vesicles move through the cell, and pull replicated chromosomes to opposite ends of a dividing cell. Like microfilaments, microtubules can disassemble and reform quickly. Figure 4.25. Microtubules are hollow. Their walls consist of 13 polymerized dimers of alpha-tubulin and beta-tubulin, right image. The left image shows the tube's molecular structure. Microtubules are also the structural elements of flagella, cilia, and centrioles. The latter are the centrosome's two perpendicular bodies. In animal cells, the centrosome is the microtubule organizing center. In eukaryotic cells, flagella and cilia are quite different structurally from their counterparts in prokaryotes, as we discuss below. Flagella and cilia. The flagella, singular equals flagellum, are long, hair-like structures that extend from the plasma membrane and enable an entire cell to move, for example, sperm, euglena, and some prokaryotes. When present, the cell has just one flagellum or a few flagella. However, when cilia, singular equals cilium, are present, many of them extend along the plasma membrane's entire surface. They are short, hair-like structures that move entire cells, such as paramecia, or substances along the cell's outer surface. For example, the cilia of cells lining the fallopian tubes that move the ovum toward the uterus, or cilia lining the cells of the respiratory tract that trap particulate matter and move it toward your nostrils. Despite their differences in length and number, flagella and cilia share a common structural arrangement of microtubules called a 9+2 array. This is an appropriate name because a single flagellum or cilium is made of a ring of 9 microtubule doublets, surrounding a single microtubule doublet in the center, figure 4.26. 4.5. The cytoskeleton 115 figure 4.26. This transmission electron micrograph of two flagella shows the microtubules 9+2 array. 9 microtubule doublets surround a single microtubule doublet. Credit. Modification of work by Dartmouth Electron Microscope Facility, Dartmouth College. Scale bar data from Matt Russell. You have now completed a broad survey of prokaryotic and eukaryotic cell components. For a summary of cellular components in prokaryotic and eukaryotic cells, see Table 4.1.